Thanks so much, Janice. And hello, everyone. My name is Jenny, and welcome to our webinar today, Illumina Sequencing, How to Plan Your First Sequencing Project. Today, we will discuss the comprehensive workflow for planning your first sequencing project, including upfront considerations with an overview of the complete sequencing process, library preparation, methods, sequencing, and data analysis. This is going to be an overview of many topics, so I will include helpful resources along the way for more in-depth information. Upfront considerations. We will start with our nucleic acid of interest. This can be DNA, RNA, or both. We then need to prepare the nucleic acid into a library. This will make the nucleic acid compatible with sequencing on an Illumina sequencer. The third step is sequencing. This is where we will read the bases from the library fragments. The fourth step is data analysis, and we want to make sure that we have a plan for the full workflow, including the data analysis, before we start any of this process in the lab. Our first step is to determine our sample type of interest, as this will affect how we prepare the libraries, sequence the samples, and analyze the data. Some examples are genomic DNA, cell-free DNA, total RNA, metagenomic samples, DNA or RNA from formalin-fixed paraffin-embedded tissues, immunoprecipitated DNA, and many others. We can sequence a vast array of different sample types, though no matter where the nucleic acid is from, we want to make sure that the extraction method we use results in highly pure samples, free of inhibitors that could affect subsequent steps. Additionally, we'll need to know how much material we have and if it's degraded, as these factors can influence library preparation and sequencing. We have some resources here: an introduction to Illumina Next Generation Sequencing, a bulletin which gives information on DNA and RNA isolation, and overviews of DNA, RNA, and FFPE sequencing. Now let's proceed to library preparation. Here we will discuss library preparation methods, how to choose the right library prep method for your project, the steps in library preparation, library quantification and QC, library preparation resources, and library pooling. There are different library preparation options depending on our project goals. Libraries can be prepared from genomic DNA for resequencing applications, as well as for sequencing a genome that has never been sequenced before, de novo sequencing. Libraries can be prepared from RNA. This can include mRNA sequencing, total RNA sequencing, or small RNA sequencing. Libraries can be prepared from metagenomic samples, so there will be multiple species present within each sample. We can create bisulfite converted libraries, which allow us to study DNA methylation patterns. For some applications, we might need a more custom approach, and these are usually found in publications. Libraries can also be targeted. If we're interested in a subset of the genome or transcriptome, whole genome sequencing or total RNA sequencing gives us way more information than we need. We can use targeted approaches to select just the genes or areas we're interested in for our project. And we can prepare targeted libraries from DNA, RNA, or both. There are two primary methods for targeted library preparation. The first is an Amplicon-based approach. With an Amplicon-based library prep, we selectively amplify only the regions we are interested in from a nucleic acid sample, and we prepare libraries from these targets. Another option is an enrichment-based approach. This is where we create whole genome or total RNA libraries, and then we use probes to target only our genes of interest for sequencing. This is helpful for larger targeted panels and even whole exome sequencing. And there are also custom targeted approaches as well. Now, how do we choose the library preparation method we will use? One resource is the Library Prep and Array Kit Selector tool on the Illumina website. This tool allows us to filter based on our project aims and interests and suggest suitable library preparation methods for us. We can use the area filter to choose our research area, like microbial genomics or cancer research. We can filter based on our application, like whole genome or gene expression. We can choose our methods, like methylation sequencing or exome sequencing. And we can also filter based on the species we're sequencing or the sequencer we're planning to use, the system. We can choose as many filters as we would like, and the tool will let us know how many library prep methods meet our criteria, then list them out to give us additional information on each option. Additional resources for choosing the best library preparation method are the following. The Sequencing Method Explorer, which has helpful information and guidance for over 200 sequencing methods. The Gene Panel Finder. This tool lets us input our genes of interest, and it will suggest compatible targeted library preparation methods that include these genes. We can use Design Studio to design custom sequencing panels, and your sales team is a great resource, not only for selecting library preparation workflows, but for experimental planning from start to finish. You can reach them at the email and phone options here, and you can also reach out to tech support for assistance reaching your local sales team. Now that we have selected the library preparation method we would like to use, let's take a look at an overview of the library preparation steps. Depending on the type of library we're preparing, some of these steps may be a little bit different. In this example, we are preparing a whole genome library. For this, we start with our nucleic acid, and in this case, it's DNA. We will fragment the DNA to ensure the length of the DNA is compatible with sequencing. We will then add adapters to the DNA and amplify the libraries. The final library looks like this schematic here. The adapters are very important and they are required for sequencing. So for the adapters, we have the P5 and P7 sequences at the very ends of the adapters. These are the sequences that allow the libraries to bind within the sequencer. Internal to that, we have the sequencing primer binding sites, which touch the DNA inserts, and these are required for us to generate data from the libraries. The index sequences allow us to identify which data are from each sample that we sequence. The index sequence for each library must be unique, and these sequences will be read during the sequencing run. We might have some additional steps like cDNA synthesis for RNA-seq libraries or targeting steps if we're preparing Amplicon or enrichment libraries. If you are using an Illumina library preparation kit, the libraries you prepare will have all of the necessary adapter sequences and will be compatible with Illumina sequencers. Here we see how the adapters are used during sequencing. The red and the dark blue fragments at the very ends of the library, the P5 and P7 sequences, are critical for binding to the flow cell within the sequencer. The light and dark green regions are the index sequences, which help us to identify the data for each sample. The light blue and orange regions are for sequencing primer binding, which are required for reading the insert and the index sequences. This is an example image, so some sequencing instruments do prime differently. If you have any questions regarding the specific sequencer that you're using, please feel free to reach out to technical support. Once we complete the library preparation, we will want to quantify and QCR libraries before sequencing. We need to have an accurate quantification and size information, as this will help us to make sure that we sequence the libraries properly. Accurate library quantification is absolutely critical for successful sequencing. Precise quantification is necessary to ensure that the correct amount of sequencing data goes to all the samples and that the sequencing output and sequencing quality are high. Each library preparation workflow will have recommendations for quantification, typically qPCR or a fluorometric method like Qubit or picogreen. We do not recommend UV spec-based methods, as these are not accurate enough for sequencing. For QC, Instruments like the bioanalyzer and fragment analyzer allow us to visualize the size distributions of the libraries in displays called electropherograms, which we usually just call traces. Traces give us information on the library size distribution and the average fragment length, and can help us to verify that the library preparation was successful before sequencing. Traces can also help us to identify if there were any issues during the prep too. This is an example trace here. For some library preparation methods, gels can alternatively be run to visualize the libraries. We periodically have a webinar for library QC and troubleshooting with the bioanalyzer, and this webinar is super informative. I definitely encourage that you attend if you would like any additional information on traces and reviewing them. And then, of course, if you ever have any questions about library traces or would like to review them together, you can always reach out to technical support. For each library preparation kit, we have a support page. The reference guide contains the full protocol as well as quantification and QC recommendations. The support page also includes reagent information in the contents and storage section at the top, additional documentation, and frequently asked questions and answers about the workflow. Once our libraries are complete and we've quantified and QC'd our libraries, we can proceed with pooling the libraries together. Pooling multiple libraries together for sequencing is also called multiplexing. Usually, we'll pool multiple samples together in one sequencing run. Sequencing runs produce more sequencing reads than we need for a single sample, so we pool the libraries together, and the data will be distributed across all of the libraries. The number of libraries we want to pull together for a run will depend on the amount of data that we want for each library. The fewer libraries pulled together, the higher the number of sequencing reads for each library. As an example, if we pull 2 libraries together, each will get about 50% of the data from the run. If we pool 50 libraries together, each library will get about 2% of the data from the run. We have resources here that will help us to determine the number of samples we want to pull together, the coverage calculator, and the library prep and array kit selector. This is a sequencing coverage calculator tool. So here we'll go through just a quick example calculation. So for this, let's say we are planning to do whole genome sequencing. So I'll select DNA application. Then the tool will bring us to the next page. And here we select our specific application from the drop-down menu. Here I'm selecting whole genome sequencing at the top. This drop-down also lists targeted panels as well as custom options. The tool will then bring us to this page. And so here we need to enter the coverage level that we want. Coverage is the way that we measure sequencing depth for a sample. 1x coverage for a genome means that each base in the genome is sequenced one time on average. 500x coverage means that each base is sequenced 500 times on average. For this application, I'm choosing 30x coverage, which is a common coverage for whole genome resequencing. If you're unsure about the coverage that you need, it is best to consult the literature in your field for the most appropriate recommendations. Then we enter a duplicate percentage, which depends on the library preparation method used, and here I'm just leaving the default value. Next, we enter the genome or target region size. For this, I'm sequencing a 120 megabase genome. Then we'll enter the read length. This is the number of base pairs or cycles that will be sequenced from each library fragment. Each Illumina library preparation kit has a recommended number of cycles. Last thing, we choose a sequencer that we want to use. And we can select multiple sequencers here to compare them as well. And for this, I'm going to choose the NextSeq 500 550. We submit, and the tool will then calculate the number of samples that can be pulled per run. With these settings, I can pull 32 libraries in a NextSeq high output run, or 10 libraries in a NextSeq mid output run. We can also check for pooling recommendations in the library prep and array kit selector. We can navigate to the kit that we're using and then click Details, the menu will open and we can expand the Specifications and Project Recommendations section. We can scroll down, and in the Project Recommendations section, we'll have pooling and read length recommendations. Once we determine the number of samples we can pool together, we can start pooling. So we want the data in the sequencing run to be distributed evenly across all of our samples. For even coverage, we will first want to normalize the libraries. This process brings all of the libraries to the same concentration. Some library preparation kits include reagents or have mechanisms that normalize the samples for us. For other workflows, we will normalize the libraries by hand, and we call this manual normalization. Integrated normalization refers to library preparation workflows that normalize the libraries for us. Different library prep methods will use different strategies for this. Some will normalize the samples based on meeting certain input thresholds, and other preparation kits might use additional reagents and protocol steps to normalize the libraries. Not all library preparation methods include integrated normalization, and some integrated normalization methods are not compatible with all sequencers. In manual normalization, we can calculate the molarities for all of our samples and dilute the samples to the same concentration. So first, we calculate the molarities using the concentrations in nanograms per microliter and the average library sizes determined during library QC using the equation on the slide here. After all the molarities have been calculated, we can make the dilutions of each library, bringing them all to the same concentration. We can use 10 millimolar tris buffer, resuspension buffer from Illumina kits, which is also called RSB, or even just nuclease-free water for these dilutions. The concentration we want to normalize to will depend on the library yields and the sequencer that we'll be using. Once all the libraries are diluted to the same molarity, we will pool equal volumes of each one. And this will help us to have the same amount of each library in the pool. When pooling samples, we recommend pooling at least 5 microliters of each library. Pipetting small volumes can lead to inaccuracies, so using at least 5 microliters for each sample is super helpful. And then if you're using 5 microliters of every library, you might end up with a pretty large pool and perhaps more than is needed for sequencing, and that is totally fine. Any additional pool can be stored at -20 degrees. I've included links to 3 bulletins which give information on library quantification, QC, normalization, and pooling. We have a pooling calculator at the final link, which gives us volumes to use for manually normalizing and pooling the libraries. We have additional information on pooling and coverage available at the links here. Now that our libraries are prepared and pooled, we'll go ahead and proceed with sequencing. We will discuss where to sequence, how to choose the appropriate sequencer for your project, how to set up a sequencing run, and resources for sequencing and instruments. Our first question is where to sequence. You may have the sequencer you would like to use in your lab, and we will discuss more about sequencing within your lab in this section. You may instead want to use a sequencing service or core facility. These facilities may be academic or non-academic providers, and each facility will have information on how to submit samples for sequencing. Many core facilities also offer library preparation services. If you are looking for a sequencing service provider, we have a list of providers that have been certified by Illumina. The link here gives more information on Illumina Propel certified providers. So next, we need to choose the right sequencer to use. Different sequencers give different amounts of data. We'll need to know how much data we need per sample and how many samples we will sequence to determine the best sequencer for the project. If you are submitting your samples for sequencing, core facilities will likely give sequencer recommendations as well. Each sequencer has a specific data output. Benchtop sequencers, like the iSeq, MiniSeq, and MiSeq, are typically used for sequencing smaller genomes and targeted panels, which don't really need as much data. The NextSeqs give more data and are more appropriate for higher sample numbers and applications like RNA-seq, exome sequencing, and ChIP-seq. The highest output instruments are the HiSeq series and the NovaSeq. These instruments are suitable for large genomes, transcriptomes, and exome sequencing, as well as pulling together high sample numbers. We have a sequencing platform comparison tool, which is a helpful resource for deciding on the most appropriate sequencer for any project. Once we have decided on the sequencer that we will use, we will plan the run configuration, prepare the sequencing consumables, denature and dilute the library, and load the libraries and sequencing reagents into the sequencer. If you have submitted the libraries to a core facility for sequencing, you may or may not plan the run configuration, and these last 3 steps will be done at the sequencing facility. To plan the run configuration, we will decide on the read lengths. An example of a read length is 2 by 150 or paired 150 base pair reads, and each Illumina library preparation method has read length recommendations. Next, we will need to list out our library and index information. This will help us to identify all of the samples pulled together according to the index sequences added during library preparation. We may also specify some post-sequencing data analysis options at this stage. Depending on the sequencer you are using, you may use one of these 4 options for configuring the run: Illumina Experiment Manager, Local Run Manager, Base Prep Tab, or Base Instrument Run Setup. Depending on the library preparation workflow, we may also have run setup templates available on the library preparation support page of the Illumina website. We will go ahead and take a closer look at 2 of these options today: Illumina Experiment Manager and Local Run Manager. Illumina Experiment Manager is a software that you can download from the Illumina website, and I will include a link for this a little bit later. This is the first screen that we see when the software is launched. We have a few different options to start, but for now I'm going to select Create Sample Sheet. On the next screen, we can choose a sequencer we're planning to use. So here I'm choosing the MiSeq. And then next, now we can select an analysis option. This will determine the type of analysis that will be performed once the sequencing run completes. Here I am selecting Other than FASTQ only. We will discuss more about what this means a little bit later in the presentation. Now, we will enter the run configuration information. The first field we need to complete is the reagent cartridge barcode field. If you know the barcode of the sequencing reagent cartridge you will use, you can enter it here. If you do not know the barcode, you can enter a value that will help you to easily identify the sample sheet, as this field will be used to name the sample sheet file. We can then choose the library preparation workflow and the index adapters that we use for the libraries that we're sequencing. Next, we'll select the number of index rates for the run. We can enter the experiment name for the run and the investigator name and the description field. These are optional. We then select the type of run we'd like to do, single read or paired ends, and the number of cycles for each read, so how many bases will be read in each read. On the right-hand side, there are workflow-specific settings. This section will fill by default based on the analysis chosen, but these options can be changed if needed. Then we'll go ahead and select Next. This will bring us to a page where we can enter our sample and index information for the libraries in the pool. Once we've added unique sample IDs and indexes for all of the libraries, the sample sheet will give a green valid status near the bottom, and then we can select Finish. Illumina Experiment Manager will then ask you where you'd like to save the sample sheet, and will ask you if you'd like to view the sample sheet in Excel. You don't need to view it in Excel, but I will here so we can take a look. The sample sheet is a .csv file, so a comma-separated value file. And when we view the file in Excel, we can see the workflow settings that we input in the Illumina Experiment Manager software. This includes the instrument we're using, the read lengths, and the sample information. If the sample sheet was created on an off-instrument computer, we can transfer the file to the sequencer. Now, we will review setting up a run configuration with Local Run Manager. With Local Run Manager, our first step is to select Create Run from the top right, and then select the analysis workflow we want to use. For this run, I will choose Generate FastQ. Next, we will give the run a unique run name, and then we can enter an optional run description if we'd like. We'll select the library preparation indexes we used from the Library Prep Kit drop-down menu and select the number of index reads. We'll then select the run type, single read or paired end, and fill in the read lengths we'd like to use. We can then scroll down the page to the sample table. And this is where we enter in the sample IDs and the index information for the libraries in the pool that we're sequencing. And once all of our samples are added, we can select Save Run. Now we can see the run that we've created in our list of runs ready for sequencing. More information on Illumina Experiment Manager, Local Run Manager, and Base Space Prep tabs can be found at the links here. So now we will go ahead and prepare the sequencing reagents. We will thaw any frozen components first. We have thawing recommendations in the system guide for each sequencer. We will then move the flow cell out of 4-degree storage and bring it to room temperature. We have a bulletin that gives information on how to thaw and store sequencing reagents for optimal performance, and I will include a link to that in just a few slides. Our next step is to denature and dilute our library pool. Libraries must be single-stranded for sequencing, and each instrument has a guide for denaturing and diluting the libraries. For most sequencers, you will need to denature the libraries before loading them onto the sequencer. That said, some instruments will do the denaturation step for you. For denaturation, we use sodium hydroxide, though sometimes we will do a second heat denature step as well. Just before loading the libraries, we dilute them to a final concentration that is compatible with the sequencer that we're using. These concentrations are also given in the denature and dilution guides. We may add the PhyX control library to the pooled sample libraries as well. PhyX is a whole genome library prepared from a viral genome. And if PhyX is included in the sequencing run, we can calculate some additional run metrics. This workflow does vary a bit from sequencer to sequencer, so it is super important to follow the steps outlined in the guide for the specific platform that we're using. Next, we'll go ahead and load our libraries and consumables into the sequencer. For most sequencers, we will add the libraries into the reagent cartridge in a dedicated position. The instrument control software will walk us through loading the flow cell, the reagent cartridge, the buffer, and emptying the waste container. Once the sequencer is loaded, we will start the pre-run checks to evaluate the sequencer. After the pre-run checks pass, we start the sequencing run. For more information on how Illumina sequencing works, we have an overview of Illumina sequencing by synthesis technology here, as well as a video which shows the full process. We have a comparison of the sequencing steps across the different instruments at the third link, and the bulletin I mentioned for thawing and storage of sequencing reagents can be found at the final link. Now that we've started the sequencing run, we can monitor the progress and review the performance. We can monitor the run in real time online in BaseSpace. We can also review the performance with the Sequence Analysis Viewer software. When we're reviewing a run, we want to check to see that the run generated the expected amount of data and that the data quality is high. Each instrument and run configuration has specifications for data output and data quality. Let's take a quick look at instrument resources. If you navigate to illumina.com, you can select Products near the top, then Instruments, then Sequencing Platforms. Then you can toggle between the benchtop sequencers and the production scale sequencers to find the platform that you're interested in. In the following slides, I will show the iSeq instrument pages. Once selecting the sequencer, it will bring you to the instrument landing page, which includes a ton of helpful resources. So first, let's select the specifications option at the top. The specifications here are the values that we want to use to evaluate the run performance. We have information on the output we can expect, as well as the data quality. And the Q30 metric is how we measure quality. We can go back to the main instrument page, and now let's select the support option near the top right. This will bring us to the instrument support page. This page has information and just so many helpful resources. Once the sequencing run completes, we can go ahead and move on to data analysis. We will discuss FASTQ generation and demultiplexing, next steps in data analysis, software options for data analysis, and data analysis resources. Our first steps are FASTQ generation and demultiplexing. In FASTQ generation, the raw sequencing data is converted into A's, T's, C's, and G's. The first line for each entry in a FASTQ file is an identifier for a read. Then, the FASTQ file includes a strings of bases given from each library fragment, along with the quality scores for each base. Here, we can see an image of the bases in one read, and following the plus sign, the separator, we have the quality scores. FASTQ files for each sample may contain hundreds, thousands, or millions of reads. At this step, we also demultiplex the data, which means that we separate out the data for each of the libraries that were pooled together for the run. The FASTQ files for each sample will list all of the reads generated in the sequencing run that correspond to the index sequences that we added during library preparation and sequenced during the run. For runs on the iSeq, MySeq, MiniSeq, and NextSeq 500, 550 runs set up with Local Run Manager, along with any run set to upload to BaseSpace, the FASTQ generation will happen automatically once the sequencing run completes. For NextSeq 500, 550 runs without Local Run Manager, as well as HiSeq and NovaSeq offline runs, we will need to manually initiate the FASTQ generation and demultiplexing process with the BCL to FASTQ software. For the NextSeq 1000, 2000, there are both on-instrument and online options for FASTQ generation and demultiplexing. Now we can further analyze the FASTQ files. The downstream analysis will depend on how we want to apply the data. We might do de novo assembly for a genome that was sequenced for the first time. For resequencing, we will align the reads to reference sequences, like a reference genome. Then after alignment, we may want to do variant calling to find SNPs and indels present in the samples. We can also do genotyping at this step. We may want to do counting for applications like RNA differential gene expression. This is not a definitive list at all, and there are many analysis options. For Illumina library prep workflows, we can give guidance on appropriate analyses to use. Local Run Manager is a no-cost software that can be downloaded and used for data analysis of iSeq, MiniSeq, MySeq, and NextSeq 500, 550 data. These are the analysis modules currently available within Local Run Manager. MySeq Reporter is available on the MySeq and can be run on off-instrument computers as well for the analysis of MySeq runs. These are the analysis workflows available within MySeq Reporter. BaseSpace has many analysis apps that can be used for data analysis online. Some of these apps are created by Illumina, and some are created by other research groups to provide data analysis options for many sequencing applications. This slide shows just a fraction of the available apps on BaseSpace. I also want to call attention to the Dragon analysis workflows. We offer several Dragon analysis apps in BaseSpace, as well as offline server-based options. At the first link here, you can access our recorded webinar that gives an introduction to sequencing data analysis. We also have public data within BaseSpace. And public data includes example analyses as well, which can be super helpful for experimental planning and selecting analysis workflows to use. We also have links for analysis options we've discussed today. Today, we've discussed upfront considerations for sequencing, including recommendations for input nucleic acids, the purpose of library preparation, different methods for library preparation, and how to determine the most appropriate method to use, the sequencing workflow, and how to choose the right sequencer for each project, and analysis options and methods. We've discussed resources for learning more about Illumina Next Generation sequencing and tools to help plan our sequencing projects. Additional technical support webinars can be found at the first link. We have upcoming webinars, and we can register for these here, and links to recorded webinars on multiple topics from library preparation to data analysis, which can be viewed at any time. We have helpful support bulletins, which include information on best practices for library preparation, sequencing, and data analysis, along with many other topics. Finally, we have publication reviews, which feature applications of Illumina Next Generation sequencing technology. We have a beginner's guide to NGS page, and you can always reach out to technical support via techsupport@illumina.com or by calling us directly at the number here. Thank you so much for taking the time to join me. I hope that this has been helpful for you.